Okay, very good evening here. Hopefully, we are doing fine. Let's get straight into our lecture for today. It's about proteins. And previously, we have concluded with carbohydrates, and we are getting into understanding of the next nutrient called the protein. And to begin with, we need to first understand here that at the end of this lecture, I will actually want you to have understood the process of digestion, the role of various enzymes and the secretory juices in your digestion. So meaning at this case you should be able to describe the process of protein digestion step by step, indicating clearly the various enzymes and the secretory juices that are involved. So for this case, let's go ahead. We are going to explore also in length how amino acids can be removed from the body while replenishing energy. That's actually what you call the alanine cycle. How are amino acids removed from your body and then used for energy? Because first, your body does not really have the ability to store amino acids. It has no amino acid store. It has just an amino acid pool. We'll get to understand the difference. A storage is which doesn't exist for proteins in your body, but we have a pool. You get into length about that amino acid pool. But what I mean here is that we are going to explore how amino acids can be removed from your body while replenishing your energy. So you remove the amino acids while you get energy from it. It's something that is so much beneficial, and we'll get to understand that in depth. Then you understand the concept of amino acid pool, nitrogen disposal. Yes, so these are some of the things that we are going to also understand in this lecture today. With many more that will come along as we discuss through the lecture. And to continue, we can get into the process of digestion. And first, we need to understand that in the process of digestion, there is a need for proteolytic enzymes. There is need for proteolytic enzymes, which are responsible for the digestion of these proteins in your body. And these are produced in the stomach. We will know which ones are produced from the stomach, which ones are produced from the juice, from the pancreas, and which ones from the ileum. I think each one of these sites, I think, have also have their juice, like the gastric juice from the stomach, pancreatic juice from the pancreas, also, which are also coming from there. So let's continue. Let's continue here and understand that these amino acids or protein have been these proteins here have been built by bonding them together, and this bonding here is done in a way that one amino acid is bonded to another. So what happens is that we get them hydrolyzed, and the peptide bonds here are hydrolyzed by peptidase enzymes. By peptidase enzymes. Okay, fine. Then these peptide bonds are hydrolyzed into individual amino acids. What you're seeing A here is amino acids, and these enzymes are called peptidases. And these peptidases right here are of different types. So one type is the endopeptidase. For them, they act inside the core of proteins, and they form small peptide fragments. So a whole protein, a big one, fully formed, is broken down into smaller fragments by what these enzymes call endopeptidase. And the second type of this peptidase is exopeptidase. This acts from the external sites, meaning one, it acts from the amino terminal or the carboxyl terminal ends of the protein. And we have this grouped into the aminopeptides. The aminopeptides here break down from the amino side, the amino group. So the amino group of an amino acid on the joining. Of course, we have a long chain, or maybe a dipeptide can be bonded together, and at the extreme end, one of these amino acid that is been bonded has an exposed amino group, and the extreme end, the other amino acid also has a carboxyl group that is exposed. So, and this exopeptidase here, the one called aminopeptidases, actually they break the amino acids, they start cleaving them from the amino group, and then the other, they act on the carboxyl group, which is called the carboxypeptidase enzymes. And beside that, we also have enzymes called zymogens. These zymogens here are the proenzymes, meaning they are produced in an inactive form. These are peptidases that are secreted in an inactive form, and when these zymogens reaches the site of action, they are activated. Oh, but why are some of these enzymes actually produced in an inactive form? That one, they protect your lining from being digested. If you have enzymes that has been produced in an active form, immediately they get to act. So they need some condition to activate them. Conditions like acidic pH, that is in the case of the stomach, that secrete our gastric juice. So we'll get to find more details about that as we continue. And these are the digestive enzymes we have here. One, in the mouth, there are no digestive enzymes for proteins, unlike for carbohydrates. So right here, the kind of digestion that takes place in the mouth is just mechanical digestion, which involves the chewing, rolling the food, mixing it with saliva, lubricating it, and then swallowing it. That passes through to the esophagus, then tail down to the stomach. And then in the stomach, we have pepsin and renin. We'll get to look into the details of these two enzymes, which are, these are the active forms, but they have their proenzymes forms, that are the inactive forms that are produced. And then we have the pancreas. Pancreas has the trypsin, chemotrypsin, elastase, carboxypeptidase A, carboxypeptidase B. And then now we have in our intestine, we have the aminopeptidase and the dipeptidase. We'll get to discuss them along the way. Then to start with the stomach, like we discussed earlier, there are no enzymes in the mouth, so we are getting to understanding what happens in the stomach. If you ask to discuss the process of digestion of proteins in the mouth, that's basically mechanical, and that's what you describe. The process of chewing it and softening it, mixing it with saliva, lubrication, and then swallowing it. That's basically all you do on the digestion in the mouth, which is basically mechanical. Yeah. To continue with us in the stomach, here is that the secretion of a hormone gastrin is stimulated, and this is secreted by the gastric mucosal cells. And this hormone gastrin here stimulates release of gastric juice. What does this contains? It contains the HCl, which is hydrochloric acid in the stomach, and it has pepsinogen. Like I said earlier, we have pepsin in the stomach, one of the hormone, one of the enzymes, sorry, not a hormone, which is produced in an inactive form. So it is a pepsinogen, which is inactive, and then it gets converted into pepsin to act on. And these are inactive form, and for infants, I think there is an enzyme called renin. Yeah, and the pH of the stomach is about 1.5 to 2.5 pH. And now HCl, what's the role of this? One, it denatures protein. It denatures proteins. These proteins can, I could simply say enzymes as well. No, sorry. This could be the germs that have been taken along with food and everything. Second, it activates our enzyme that was inactive, meaning it converts our pepsinogen to pepsin. Then antibiotics, it kills any bacteria that is around in your stomach that you've digested. So this is more like a defense, a way of defense, and it's also called a first line of defense. Then pepsinogen here is from the chief cells, from the chief cells. It is here, it is activated to pepsin, just like we said earlier, and its role is the autocatalysis of the proteins, meaning it involves in the hydrolysis of the proteins. So it breaks down proteins here. Renin, which is found only in infants, what we call chymosin, and it is only found in infants. What it does is curdling of milk. I don't know if you have seen scenarios where a baby have just breastfed, and after breastfeeding, they get a challenge and vomit it out after some few minutes. So you find out that some of the milk they vomit out or spit out have some lumps. So that is a clear indication that this enzyme called renin has already started acting on the milk, and it is converting it now into a more solid form, what we call the paracasein. So becomes calcified, and then to be hydrolyzed, and then it will be further hydrolyzed by the enzyme pepsin. So you realize after that you release all that lumps, those small lumps. That is now what we call a cadu. It's more like a clot. That is now actually what we call the paracasein. So to continue with, I think that was all for the stomach, the two enzymes. I think if we are asked about the enzymes, will be able to explain the two enzymes and the juices that have been produced from the stomach and where from. The gastric juice, and then we also need to take note of this hormone gastrin and its role in digestion. So we can look at that and what does it do. We can ask you for the regulation of digestion. That will be actually the role of these hormones like gastrin, which will stimulate the gastric juice, and those are all other things. So let's continue on to the pancreatic and intestinal enzymes. The stomach contents will pass into the duodenum. Remember, it has low pH, that is acidic, 1.5 to 2.5, around that. So what happens is that low pH that is from the food in the stomach is one that stimulates the production of two hormones, secretin and chemocystokinin. Then the secretin here stimulates the production of bicarbonate to neutralize the low pH, and the pH in the duodenum, in the duodenum is supposed to be alkaline. So this has to be neutralized to give a conducive condition for the process of digestion to continue. Cholecystokinin here stimulates the secretion of pancreatic proteases. This is the enzymes those enzymes that are responsible for breakdown of proteins in the duodenum and the rest portion of the gut. And finally, we shall have endopeptidases, which are 1-elastase, trypsin, chymotrypsin, exopeptidases, which are carboxypeptidases and aminopeptidases. So we had discussed about this earlier. So I'll get to discuss that with you guys maybe sometime back later. But I think we discussed about the exopeptidases. We talked about it briefly up there, and the endopeptidases. So we know them, and right here we are getting the examples, which are elastases, trypsin, and chymotrypsin for endopeptidases. And then the exopeptidases here, the carboxypeptidases and aminopeptidases. So we'll get to do that. And right here is that we have the endopeptidases. They are secreted in this zymogen form, which is an inactive form. So in other words, one is trypsinogen, proteoelastase, chymotrypsinogen, and now we can get the examples of endopeptidase enzymes, which are right here. And then this cleave the internal peptide bonds and results into formation of smaller peptides. So like we said earlier that these ones break them. These ones break the protein molecules into small fragments of peptides. And then I think the process of digestion will still be continued upon and acted upon by other enzymes to break the peptide bonds. And then they do that. So next enzymes, the exopeptidases. This is more a continuous repetition, but this is necessary for you to get them to sink into your form, into your head. So these cleave the one amino acid at the time. They cleave an amino acid at the time from the amine or the carboxyl terminus of the polypeptide chain. It's more like you have a long chain of amino acid, the extreme of a protein, sorry. Let me say a polypeptide chain. So at the extreme, at the other end, one end has an amino group and the other end has a carboxyl group. So these the exopeptidases here. The aminopeptidase starts cleaving from the amino group, one amino acid at a time, and then the carboxypeptidase starts cleaving one amino acid at a time from the carboxyl end or the carboxyl group of the amino acid or the polypeptide chain. So action of pancreatic enzymes, one is trypsin that has been produced in its inactive form called trypsinogen and activated into trypsin. One, it is activated by enterokinase. It's an enzyme and itself. It is specific for cleaving peptide bonds by basic amino acids. So all those basic amino acids that you eat from your food and everything are cleaved by what you call the trypsin, and it activates other zymogens. What does it do? This has to be something that you note down. That this enzyme here is very important in activating other zymogens, which are chemotrypsinogen to I think chemotrypsin, proelastase to elastase, and then carboxypeptidase to peptidases. So we'll get to know the active forms later on, and then they are activated. One is the chemotrypsin, which is chemotrypsinogen is converted to chemotrypsin, the inactive form which we had earlier talked about or we have seen on this very slide. Also, as you see where my cursor is, we have chemotrypsin. It can be activated into chemotrypsinogen activated into chemotrypsin by the enzyme trypsin. So what happens is that this enzyme chemotrypsin acts on peptide bonds that are formed by the carboxyl group of the aromatic amino acids. Then elastase here, it acts on peptide bonds that are formed by glycine, alanine, and serine. So you get to understand which amino acids are easily acted upon by these enzymes. Elastase for glycine, alanine, and serine. These are all amino acids. Let's continue into carboxypeptidases, and this does not act on dipeptides. The carboxypeptidase A is one of the kind that enzymes that are acting on the carboxyl terminopeptide bond that has been formed by tyrosine, phenylalanine, and tryptophan. Then carboxypeptidase B, and this act on the carboxyl terminopeptide formed by lysine and arginine. So you need to take keynote or note this so carefully and understand about this breakdown. Then we're diving into what we call the intestinal enzymes. What do they do? These are the products of hydrolysis, which are the pepsin. The pancreatic enzyme is completed by intestinal peptidase. So action of pepsin, and all other pancreatic enzymes, their process comes to completion in the intestine by intestinal peptidase in the ileum. So you can have maybe an MCQ, multiple choice questions, which of the above enzymes completes the process of digestion in the ileum, intestinal peptidase? Which part of the gastrointestinal tract does digestion in? So you can have all those options. So let's head straight into aminopeptidase, which are one exopeptidase. It acts on the N terminal of peptide bond to release free amino acids. That's just from the amino N. The di and tripeptidases, we have endopeptidases, which will act on dipeptides to release free amino acids. Now, let's talk about the absorption of amino acids. We have come to conclusion that the process of digestion here ends in the intestine. That's for the in the ileum. Sorry, that is for proteins, just to be specific. So majorly, let's talk about the transport absorption. How are these things absorbed after the whole process of them being broken down? Is that a majorly active transport through sodium amino acid co-transport. And this is an energy requiring process that uses the energy to move the amino acids along the transporters to reach the bloodstream. So let's get this. In adults, essentially all protein is absorbed as tripeptides, dipeptides, or amino acids. So amino acids or peptides are released into the intestinal bloodstream and transported to the liver by hepatic portal vein. So immediately after your digestion and the food being absorbed into your body and everything, the amino acids is being absorbed, it is transported straight to where the liver is for metabolism, and and then it's transported by to the liver by hepatic portal vein. So that is basically it to what happened. Then we get to understand that 50 to 65% of amino acids will remain in the liver, and they will be used to synthesize protein, nitrogen-containing compounds, and form purines and pyrimidine bases, which are nucleotides. Or in some cases, they can be used for energy. What cases are these? Lack of energy, when you're starving or you're fasting. So they get to use this for energy. Then looking also at that amino acids that leave the liver are transported to the rest of the body to be taken up and utilized by the other cells. So your body will need to feed throughout. So much as these amino acids passes and are passed and retain some of the bigger percentage also, which is 50 to 65, this been which is also been retained in the liver or utilized in the liver for the process of protein synthesis, nitrogen, of manufacture of nitrogen-containing enzyme molecules or compounds. There are also the rest of the bodies also will utilize the same amino acids. So what is passing also via the liver, the rest will also go into the rest of the other parts of the body and be utilized for other functions like growth or formation of immune system, repair of tissue cells. So that's basically it about that. Then absorption of intact protein and polypeptides. And intact protein is one that has not been fully digested or broken down. So it is just a solid that is protein form. So what happened? This occurs in infants. So if you're an adult, and this is actually one of the things that causes allergies in adults, that you cannot break down such kind of proteins. So once you take it into your body, your immune system recognizes it as foreign substances. However, in children or infants, is that they have the endocytosis or pinocytosis. So adults do not have the capacity to digest intact protein. That is what I mentioned earlier. Your body cannot break down or digest protein. Intact protein, sorry. So seems to be cause of and that seems to be the cause of most allergies if it occurs in adulthood. You're an adult, you're getting allergies when you eat proteins, then there's definitely this kind of a problem, and you need to go and get diagnosed from authentic health facilities. Protein metabolism, now moving away from actually what we call protein digestion, and diving into metabolism, is that amino acids contain nitrogen, just like we have seen in the structure here. So there's a fade of this nitrogen. Where does it end up in? So one is that this may remain on the molecule and be incorporated into the product that the cell is making. For example, a polypeptide. Polypeptide, the nitrogen may remain on the polypeptide, and then if your body is making some kind of proteins and is using the polypeptides, it will also be used along with it. Then maybe it can be transaminated. Yeah, that's true. Transamination meaning transfer of the amino groups from one amine or an amino acid to a carbon skeleton that will form a new amino acids and also a keto. It also forms a keto acids. Just like you have seen in the structure here is that we have amino acids with a keto acids. PLP, pyridoxal phosphate, the enzyme is aminotransferase step. So here they form this product and this other product. So I think that is it for this slide. And the amino group may be removed from amino acids in the process known as deamination. That is also another way. So new proteins are amino acids are recycled to make new proteins, and each time amino acids keeps being replaced and recycling and everything. And if you use it, it can be broken down still. For instance, you use it for muscle building, and then with time, your body can still break it. Your body can still break everything down, and that's the process of recycling. So all cells in your body continually break down proteins and build new ones. That's what we call protein turnover. We'll get to discuss that concept of protein turnover so soon. And there is a balance between protein synthesis and protein degradation or breakdown. Then approximately daily you have to get that approximately in a day 250 grams of protein in the body are dismantled or they are broken down, and 250 grams of the proteins are also built. That is what we call protein turnover. New proteins, amino acids are from food, and those from the protein destruction are placed into the pool, which will get the pool to understand. Amino acids for protein turnover are from the pool. Critical to maintain amino acid levels within this cellular pool by consuming high quality proteins in the diet. So as we have a pool there, I could say it's more like a bank that we store our things in. That's just an analogy I'm giving. You can have something like you can look at it as a bank. You put there everything, your money and everything, but you can also withdraw it. Meaning you have some other things that make you put there. You have a job that will help you put something. To come to understand about replenishing and the depletion of the amino acid pool, what processes add on the amino acid in the amino acid food and what processes removes them. So we'll get to discuss that so soon, and as we continue in this lecture. So your amino acids needed for building new proteins will be obtained by increasing protein distractions from other tissues within the body, especially muscles. Then the amino acids protein turnover are there from the pool. So getting into the concept of amino acid pool here, and this is the total amount of amino acids available in the body from diet, protein recycling, protein synthesis, and non-essential amino acid produced by the body. But what happened here is that there is a constant movement in all changes that occurs in this pool. That at each time the pool is being replenished meaning you've been adding more and more and more, but also more has been going out and more has been broken down, metabolized. So we generally need to be added. So these are the things that results into addition or into the amino acid pool. One is proteolysis, protein synthesis takes, and then the other amino acids also adds into the pool. Catabolism will take away from the pool, and biosynthesis of nitrogenous compounds, which includes porphyrin, creatinine, carnitine, hormones, nucleotides. All of them involves the use of nitrogen. So that's fine. So let's get into the next concept of nitrogen disposal and excretion. Nitrogen accruing, or we can say a nitrogen that is circulating outside the liver, is transported to the liver as glutamine or alanine. This glutamine here, is one of the major amino acids that is helping us in this process of protein metabolism, breaking them down and everything. Or it is also transported as alanine. You get to understand that. And then in the liver, the nitrogen is released as free ammonia. Just you reach the liver, you release free ammonia, which can then be used or broken down through the process of urea cycle or ornithine cycle, and then excreted as urine, because ammonia is a toxic compound in the body. So ammonia is incorporated into urea, which is less toxic, and the urea is released from the liver into the bloodstream and excreted through the kidney. So now if you get back your things as a waste, which is very toxic, your body will need to excrete it. And that is one thing that is done by the kidneys. As you take in a lot of your proteins and everything happens and things have been broken down, you will surely need to have also your protein being excreted in the form of urea or urine in your body. Then protein metabolism, still we are under that same topic. Much of the body is made up of protein, and that is very so true. You can see that almost the whole body made up of the proteins, and that is one of the major things. Your structure, the whole of your structure here is more of a proteins. And if you look at the joints, the tendons, and the flesh that is on your bones, those are all proteins. So they represent cell signaling receptors. Meaning these amino acids or proteins can be signaling receptors, which or signaling molecules, can be structural members. It can be enzymes, intracellular trafficking components, extracellular matrix scaffolds, ion pumps, ion channels, oxygen, and carbon dioxide transport. That is the role of hemoglobin, the oxygen and carbon dioxide transporters. So those are some of the kind of proteins that are very important in your body. And this is not even the complete list, because there are really more to that. We have the tendons, the collagen, the muscles, and everything in your body. So we get into what you call the glucose alanine cycle. And this is primarily a mechanism for skeletal muscle to eliminate nitrogen while replenishing its energy supply. What happens? Your body have to remove the nitrogen, the excess nitrogen that comes from your proteins, or the breakdown of these proteins, and then they replenish, and they do this in a way that they replace some energy. So the pyruvate from glucose oxidation is transaminated to alanine, catalyzed by alanine transaminase. That's a transamination reaction. Hopefully I've put some visuals there that will be able to understand. Then the transamination reactions requires an amino acid donor, which is called a glutamate. Alanine then enters the bloodstream and is transported into the liver. Then in the liver, the alanine is deaminated. Deamination meaning you remove the amine group and then it forms for your pyruvate back. Meaning you have recycled back your pyruvate. So the nitrogen released will be used for urea synthesis. And now the pyruvate that is formed to be used for gluconeogenesis. And I hope I will be able to remind you about gluconeogenesis, one of the lectures we had under carbohydrates. I think that has been discussed, so there is really not much for me to go through it so much. The vessel is right here, the glucose-alanine cycle. It occurs in the way that this amino acid glutamate here reacts with pyruvate to form alanine and alpha ketoglutarate. What happens is that these amino acids incorporated into the Krebs cycle, they form alanine. Alanine will diffuse into the bloodstream and then right into the liver and then undergoes the process of gluconeogenesis until it forms glucose. We know very well gluconeogenesis, formation of glucose from the body. So friends, this is today the end of our lecture, and I want to say thank you very much for listening.